Hello and welcome to this little session where we're talking about exosome therapeutics or extracellular vesicle therapeutics from academic research perspective. My name is Lötvall. I come from the University of Gothenburg in Sweden. I've been working on extracellular vesicles for almost 20 years now. It's been quite a journey, and now we're approaching a time when the extracellular vesicles are coming to the clinic. So with me today I have two prominent and experienced extracellular vesicle or exosome researchers. First, Kiang Lim from Singapore. Briefly introduce yourself, please.
Hello, I'm Lim Kiang. I'm from Singapore. I've been working on MSc exosomes for as long as young Lötvall has been working on EVs, almost 20 years. And I'm basically an academic scientist.
And we should also point out that you have taken mesenchymal stem cell exosomes to the clinic in Singapore.
Right, so we have done one phase 1 safety trial for MSc exosomes to treat psoriasis. And we completed it last year with no adverse events.
No bad events. And also we have Kevin Hicok, who is a biologist and process scientist, I would say, who has also got experience of extracellular vesicle manufacturing for clinical therapeutics. Briefly introduce yourself.
Kevin Hicok. I started my career at Mayo Clinic and have spent the last 30 years in the mesenchymal stem stromal cell space developing therapeutics for these cell therapies, and more recently joined a biotech company in the extracellular vesicle space, and we were able to drive progression of that particular EV product through phase 3 clinical trials, and those trials are still ongoing. But so far, the profiles for those EVs have been excellent, both from safety and efficacy perspectives.
And we should say you began by treating cytokine storm, I guess, in COVID-19.
That's right. During COVID, we pivoted our indications toward acute respiratory distress and saw dramatic results in our initial studies.
So today we have visited Exosome Bio and their research and manufacturing facilities that are in two different places. So I would like to ask Dr. Lim, what were your impressions of the manufacturing facility?
I've visited several in the last couple of years, and clearly the Exosome Bio facility is one of the most advanced that I've seen so far. Definitely very impressed with the computerized systems that they have in place to monitor the processes over a 24/7 type of time cycle. Very impressive.
So, Kevin, what's your reactions to the facility?
Yeah, similarly to Dr. Lim, I've seen a number of these facilities over the last couple of years, and this is really impressive. I would say that it was money well spent. The design of it is perfectly compliant with CGMP regulations for manufacturing, and I think they've got a good step forward in the field for having a really excellent state-of-the-art manufacturing site.
Yeah, so the importance for GMP is, of course, that the vesicles that are developed for clinical use are as effective and as safe as possible. And for that, you need to have a GMP manufacturing process in place. And I've also been to other facilities. They're usually smaller and simpler, maybe. And this is, I think it's quite unique that I don't think there's any facility like this anywhere in the world that is focused on exosomes.
Certainly not by individual companies that are EV companies. Most companies use CDMOs or CMOs.
There are other big companies that are doing, but not pharmaceutical. Yeah. So Exosome Bio are developing their exosomes for skin applications. And there are many types of exosomes from different sources, from blood, from plants, from animals, and so on. What's your view? What is the best type of extracellular vesicle? So the most appropriate vesicle, I would say, to use for clinical therapeutics, for anti-inflammatory clinical?
Right. So if we think of EVs as communication vehicles, then you would think that it has to communicate with something they are similar with. They have to talk the same language. So in my opinion, human EVs for human cells should be the most appropriate communication means. But because the EVs across the animal species carry proteins that are conserved, so EVs from various animals could still potentially talk to each other, so they may still work. But I think the best would be to use human for human EVs for human cells.
I have a comment on that as well. Kevin?
Regarding species, I agree with Dr. Lim about using human EVs. I think that makes a lot of sense. They've been evolved over many, many years to serve that purpose, to communicate between cells. And I think the mesenchymal sourced EVs have a lot of potential. The cells themselves have been shown to modulate inflammation, and it's their secretome and these EVs that seem to be able to do that. So I think there's really robust evidence in the literature that support using those kinds of EVs for mediating inflammation.
So there's been a trend to test or evaluate whether animal-derived vesicles, like cow serum, basically derived exosomes, could be useful. I would be worried personally as a clinical allergist. So I'm an allergist, as I mentioned in the beginning, and giving non-human material to the skin could definitely induce allergic sensitization, which is absolutely not anything we want. So for stability and for functionality, what are the methods? After the vesicles have been harvested from the big cell cultures and so on, I'll start with you, Kevin. What are the best way, you think, to transport those to the patients and to apply them to the patients?
Yeah. So depending on the purity of the product, whether it's really purified proteins or EVs, or whether it's more of a secretome-based therapeutic, there's going to be potentially some bioactive molecules that could degrade the product. So classically, people have frozen the product in ultra cold temperatures until use in the clinic. That presents a cold supply chain issue, of course, and so a number of companies have moved toward lyophilization and stabilization of the EVs in a dehydrated state, which I actually think holds a lot of promise. And I know of a number of companies that are trying to develop these successful lyophilization methods for storing the EVs at room temperature.
So just quickly to explain what lyophilization is, it's basically drying the material so it becomes a powder, more or less.
Yeah.
And then before you apply it to the patients, you add liquid. I think a lot of stability data and maintained mechanistic function will need to be tested and evaluated for that to really work. Dr. Lim?
I agree with Kelvin about this lyophilization being the best way forward, and my group has been working on that for several years. We had a patent application in the process. So we routinely lyophilize our exosomes, and we have shown that they are very stable. So we observe that they are stable at 30 degrees, 40 degrees for up to 3 months easily. So, and it's easy, like you say, in terms of transport and storage.
I can say that when we take bacterial vesicles and lyophilize them, dry them, we can keep them in room temperature for quite a long period of time, several weeks or months, and they maintain functionality. In my lab, we haven't done that yet with mesenchymal stem cell vesicles. Have you done that?
So for clinical trial, our exosomes was in a ointment formulation, and it can be kept at room temperature. So we have done stability, I think the last was 12 months. At 12 months, 30 degrees, it was still stable.
Quite stable, that's impressive. So I'll continue with you. What's your vision about exosome therapeutics for the short term and the long term?
I think, like Kevin said, it's a very, very promising drug. I think one of the big challenge is in the regulatory perspective. I think because it's so complex, the regulation will be very challenging. But having said that, I think the community as a whole has been very cognizant of this challenge, and we have been discussing a lot and publishing papers. I think it's tractable once we recognize that it's complex and the regulation needs to reflect that and understand that. And I think the biggest challenge of all is understanding how it works. I think the general talk outside of the community is that, oh, this is another MSC, nobody knows how it works. And I like to correct that misconception. I think we are now, most of us recognize that this will stop us from getting clinic, and we have been working hard drilling down to the molecules so that we understand which molecules in the EV works, how it works. We are not there yet, but the data that is coming out from various labs is very promising. It's going to be very rigorous, I think, more rigorous than what we have seen in the MSC world.
Yeah, I agree. I think what I'm really excited about is with the onset and development of artificial intelligence and big data sets now that the EV community are sharing with each other, that we now have the opportunity to use some serious computing power to try to understand those hundreds or thousands of interactions that are going on, whereas somebody sitting in their lab late at night trying to look at all those things gets very buggy-eyed very quickly. So I think there's a lot of potential for us to crack the nut, as they would say, regarding the mechanisms of action.
Yeah, so I also believe there's a huge future for extracellular vesicle therapeutics beyond mesenchymal stem cells. Mesenchymal stem cells vesicles can be further enhanced by loading of different molecules, and maybe that's important to really get that breakthrough in the clinic. But other types of vesicles can be engineered to carry certain molecules or deliver certain molecules to recipient cells, which I think is a big opportunity. I believe that exosome therapeutic candidates will continue to evolve, and there will be new ones for many years to come and far beyond my lifetime and probably your lifetimes as well. And with that, I would like to say thank you for your attention.